Hi everyone, Biology with Zhangshuan here. So today we are going to do AS Biology, the 9700, Cell Structure, Chapter 1.1, the Microscope in Cell Studies. So whether or not you are doing your revision or this is the first time stepping into A levels, um, I hope you are prepared because this is definitely one jump from your IGCSEs or your O levels that you have done probably in the past few months or a year. All right, so let's start off with today's topic, which is the microscope in cell studies. So for today, we will take this chapter and we separate into two parts. The first part, we are going to discuss the microscope in cell studies. So this will be the whole thing we are revolving today. For the next video, we are going to cover the cells as the basic units of living organisms. So each subtopic has quite a lengthy content and we will require more time to have a more useful discussions. Okay. So let's start off with today. The outcomes for today is to calculate magnification for images and actual sizes of specimens for drawings, photomicrographs, and electron micrographs with either scanning and transmission. Use an eyepiece graticule and stage micrometer to make measurements and use the appropriate units in millimeters, micrometers, and nanometers. So this will be the challenging one that if you're new to this should be a bit tough, but with practice, you definitely will understand this. And the last one is to define what is resolution and magnification and explain the differences between these terms with reference to light microscopy and electron microscopy. So let's start off with magnification. So magnification is the number of times that a real life specimen is enlarged in a microscope. So meaning if I have like a specimen here, this is from our view, but a microscope can go to even deeper. So they might be able to see some close up in a larger and enlarged view. Okay, so this is what we know as magnification. It comes from the word magnifying, where we go deeper and look in depth with the structure. So coming back to the formula you should know is image over actual. So image length divided by the actual length, and you get the magnification. And again, the rearranged formula can be either actual size equals to image size divided by the magnification, and image size equals to magnification times by the actual size. So this is a kind of repeated from if IGCSE biology that in your previous level, you have learned how to convert units between centimeter to millimeter and to micrometer. So you know that 1 centimeter is 10 millimeters, 1 millimeter equals to 1000 micrometers, 1 micrometers equals to 0.001 millimeters. So if you still do not, or maybe you have forgotten how to do magnification, I will link the IGCSE biology video that talks about magnification on top. So in A levels, now you step in further to a much even smaller unit, which is nanometers, which is represented as nm. So 1 micrometer equals to 1000 nanometers. So for example, if I have a specimen that is 7 micrometers and I want to convert it into nanometers, it's just very simple. Just times by 1000, you can get 7000 nanometers, which is very, very small. Okay. All right, now we will enter this tricky part, which is the eyepiece graticule and stage micrometers. So it in a microscope, right? Not all light microscope may have this, but they have another scale on the view that you can see, the field of view of the microscope. When you look at it, right, it's a circular view, right? And inside, depends on which microscope that you're using, they will have an eyepiece graticule scale or a stage micrometer. So It is used to measure the size of an object when it's viewed under the microscope, okay? And often it has 100 smaller division, which is known as Graticule division. Okay, so before calculation, you need to calibrate it. Calibrate means to adjust the stage micrometer so that it provides you an accurate scale and much easier to calculate. And of course, the calibrated eyepiece graticule can be used to measure the length of an object, which is the graticule division covered by the object times the magnification factor. You will get the length of the object in micrometers. Okay, so units is very important here. So it's important that one tip is to convert all of your values into micrometers so it's easy to calculate. Okay. All right, let's try to do an example, which is to calibrate the eyepiece graticule. All right, so I will try to repeat twice if you do not understand this. So listen to this first time. So in this diagram, you can see that there is two-stage micrometer division, because in the question it says that small divisions are 0.1 millimeters. What does it tell you? Between this point and this point, it is 0.1 millimeters. And from this point to this point is also 0.1 millimeters. So you want to convert 0.1 millimeters into micrometers, which is equals to 100 micrometers. Okay, now it's much easier to do. So each 100 micrometer, so now I just put this as also as 100 micrometers, it covers between this range and this range, which is actually 50 minus 10, which equals to 40. So what does it tell you is that 40 graticule division is 100 micrometers. And now the most important part, you want to find what is one graticule division. So one graticule division, which is just very simple. Take 100 divided by 40, and you're going to get 2.5 micrometer. So what this 2.5 tell you is that from this point to this point, right, it is 2.5 micrometers. And that is what you know, call it as a magnification factor. Okay, so this is called as a magnification factor. Okay, so I hope you have a little bit understanding now. So now we repeat the second time again. So this question says that the stage micrometer scale has a small divisions with a length of 0.1 millimeters. What this tells you is that between this point and this point is 0.1 millimeters. And from this point to another point is also 0.1 millimeters. So what I tell you before is that convert all of your values into micrometers so that it's easier to calculate. So 0.1 millimeters will become 100 micrometers. And now you want to find what is one graticule division. So the whole point of calibrating is to find out what is one graticule division. And between 50 to 10, sorry, it's total, there's a difference in 40. So between this point to this point, it has 40 graticule division, which is equals to 100 micrometers. So you can see 40, I have 100. So now you do your division. You want to find what's one graticule division. Is taking 100 divided by 40, you're going to get 2.5 micrometer. So this tells us that one graticule division, which is between 10 to 11, for example, is 2.5 micrometers. And that is what we also known as a magnification factor. Okay. All right. Now, after we have calibrated it, we know what is our this calculation. Now we are going to find out how are we going to find the width of the chloroplast using the magnification factor that we got. So the magnification factor we have is 2.5 micrometer. 2.5 micrometer. Okay, now the stage micrometer scale is replaced by a slide of a plant cell. What is the width of the chloroplast? Okay, so the width of the chloroplast, not the nucleus. Okay, so the chloroplast, let me just take one example. Let me take this. Okay, let me get a thinner line. I would say it's pointing to about maybe here. I just assume it will be 26, and here is about 30. So this is about 4. Okay, so 30 minus 26 is about 4. So I will take 2.5 now. So I will use the for one chloroplast. So one chloroplast, it has a width of about 4. So now you want to find what is the width of it. So you take the magnification factor, which is 2.5, you multiply by 4, and you're going to get 10 micrometer. This is the answer. So it's not difficult. It's just that you must understand how to calibrate this first. When they say which the small divisions are 0.1 millimeter, meaning from this point to this point is 0.1 millimeter, and you want to find what is one Graticule division. Okay? So another example is that now they ask you for what is the length of the nucleus in the plant cell. So using the same magnification factor, which is 2.5 micrometer. So I just put that MF, which is 2.5 micrometers. So now they ask you what is the length of the nucleus in the plant cell. So the nucleus is this thing. Okay. Now you want to find what is the length of it first. So assuming it's about here. So I take it as 67 and reach about here, which is about 75. So I take 75 minus 67. So this will be about 8, I think. 75 minus 67. Yes, it's 8. So I will take 2.5 times by 8. You get the answer about 20. Okay, so the answer, the closest one would be 25 micrometers. Okay, let's move on. So magnification, now we are talking about what is magnification and what is resolution. So magnification is the number of times that real-life specimen has been enlarged to give a larger view or image. So it is typically at times, for example, like times 100. It means that we magnify by 100 times. And in a light microscope, there will be two lenses, which is the objective lens and the eyepiece lens, which the eyepiece lens is the one that we always see has about of a times 10 magnification. So now they will ask you to find the total magnification view. So of course, the objective lens, there are times 10, there are times 40, there's even larger values also. So what we take is that we take the eyepiece lens, which is typically 10, and you multiply by the objective lens that you use. So this will tell you the total magnification that you use to view that object. Okay. Okay, now for resolution. Resolution is the ability to distinguish two separate points on an image as separate objects. So it's like you can see that when it's viewed, right, you can see that there is some distinct points that has a lining that shows that this is known as one object and then another one is known as another object. So a lower resolution will cause the image to be blurry. I will show you one image example later on, and the resolution of a microscope will limit the magnification that it can usually achieve. So what this tell you is that no matter how much that you magnify, if your image is not doesn't have a good resolution, it will still be blurry. So regardless, your resolution has to be really good, so that no matter you magnify as large as possible, you can definitely see that there is a distinct two separate points and they are known as also separate objects. Okay, and of course, in a light microscope, its resolution is limited by the light wavelength. Okay, so it's all determined by the light wavelength. It cannot be smaller than half of the wavelength of visible light. So visible light is about 400 to 600 nanometers. So meaning the light microscope can only view with a resolution of half of it, which is about 200 nanometers. Okay, and in an electron microscope, it has a higher resolution. Why is this so? Because the electrons are so small. They are smaller than the visible light wavelength. So it's so small that you can definitely have a resolution of so tiny, and it still be really clear. So it can have a resolution of 0.5 nanometers. Okay, so what is the difference between this magnification and resolution? So for this image below, you can see that this is known as a quite high magnification but a very poor resolution, because you cannot really see the distinct as two separate objects. You cannot distinguish them that there's two separate objects. But for this next image below, you can see that it has a very good high magnification and also a very good resolution. So you can see that this is one object and this is also another object. This is, you can distinguish two separate points. Okay. All right. So now we'll look at light and electron microscope. So from the word light, it means that it uses the visible light wavelength. And electron microscope is where you fire a beam of electron. Okay, so this, you do not need to know all of the labelings, but this is just the difference between how it looks like. Okay. And so a light microscope can only be used for specimens for objects larger than 200 nanometer, which we already mentioned that it should be half of the visible light wavelength. The specimens you can view, it can be either alive or it can be a dead specimen. And light microscope is very useful for observing the entire cell, very small organisms that are living, and tissues within organs, such as in leaves or in our human body, our skin. For an electron microscope, it is available, this should be electron, okay? So electron microscope can be available as known as scanning electron microscope or transmission electron microscope, which is known as SEM and TEM. So both of the microscope will fire a beam of electron directly onto the specimen, not directly. But for TEM, which is transmission, it only directly shoots the electron to the specimen itself. But SEM, the electron will bounce off on the specimen. And the specimens, of course, cannot be alive. It needs to be a dead specimen. And it is very useful for observing viruses, very small organisms, organelles that cannot be seen, like the endoplasmic reticulum, which cannot be seen by a naked eye. We have rough and smooth endoplasmic reticulum. You need to use an electron microscope to view it. And DNA, so small. So you need to use an electron microscope. So this is the difference between SEM and TEM. So you can see for TEM, right, the electron source, right, is shoot directly onto the specimen. So this is what I know as linear. They shoot linearly. But for SEM, what happens is that it bounces off. You can see it shoots down, but it bounces off. So this is what we know as it bounces off from the specimen, and they will produce like some wavelengths out. Okay, so it can be detected, something like this. Okay. So you must know what is the comparisons between a light and an electron microscope. So light microscope is usually small and it's very portable, and electron microscope is larger machines. Light microscope doesn't require a vacuum, but electron microscope, you need a vacuum so that the electrons can travel straight through. Then the light microscope, the specimen preparation can be very simple because you just put a slide and that's it, you can view it. But for electron microscope, there's a more complex process that you have to prepare before you start with your basically your observations. And of course, the light microscope can only have a maximum magnification of times 2000. And the electron microscope will be even higher value, be about times 500,000, which is actually very, very, you can see objects so small. Okay, and light microscope, they only have a maximum resolution of 200 nanometers, which you have to remember this value. And electron microscope, you must know that it has a resolution of 0.5 nanometers. And light microscope, the specimens can either be living or it can be dead. And electron microscope is always dead specimens. So you must know at least 2 to 3 differences between the light microscope and electron microscope. So both sides you have to know what is it. Okay, so this is one example of another example actually on how to use the scale bar, okay, to calculate the specimen. So what we do is that a lab technician observe bacterial cell with an electron microscope and produce the image below. So the scale bar measures in 2 centimeter in length, okay? And of course, the length of the technician image of one bacterial cell is measured at 7.6 centimeter. So what we do is that we want to calculate the actual length of the bacterial cell in the image. So we will use magnification, the formula. Okay, so IM. So we need to find out the scale bar. They say the scale bar is measured as 2 centimeter in length, which you need to convert to micrometers. So 2 centimeter will become 20 millimeter. You know that 1 centimeter equals to 10 millimeters, and you need to times by 1,000. You're going to get 20,000. This will be in micrometers. So the scale bar measures as 20,000 micrometer. And the actual size is between, can you see this one? So it shows that it has a ratio of 20,000 to 1. So the magnification will of course be times by 20,000. So I hope you understand from here, 20,000. So the first thing, you need to change the scale bar to micrometers, and you divide by the one that's provided in the image below. Okay, so now what you're going to do is that you want to substitute your value from the times 20,000 to find what is the actual size. Okay, I think this part is quite simple. So what you do is that you need to change your value of your one bacterial cell. So the length, the image length of it is 7.6 centimeter. So 7.6 centimeter, you have to convert into 76 millimeters, and then times by 1,000, you can get 76,000 micrometers. So you're going to use the actual size formula, which is basically image divided by the magnification. So image you get is 76,000 in micrometers, and you just divide by the magnification, which is 20,000, and you will get your actual length, which is at 3.8 micrometer. So this actual length of that bacteria, when they view it, is 3.8 micrometer, which is super duper small. Okay, so I hope you understand this. So first part is to modify the scale bar, and you find the magnification. Then you find what is the actual length of the specimen by using your magnification, and you definitely will get your answer. Alright, so that's it for this video. So thank you so much for watching. Any questions can be asked in the comment section below, and I will answer and respond to any of your queries. Alright, thank you so much for watching. Bye bye.